Hello everyone and welcome back to my channel where we talk everything science. My name is Aubrey Molln. I'll be your presenter today, and today's topic is pulmonary surfactant and its functions. So without further ado, let's get to it. Before we dive into our topic of pulmonary surfactant, I think it's very important that we review the primary respiratory structures that help make it possible for us to breathe in and out effectively. So if we were to direct our attention to the diagram on the left-hand side here, you can kind of see many of these structures are highlighted. So for instance, if we were to take a breath in, usually through the nose or the mouth, which is the nasal cavity, it enters our body and it travels down this windpipe, which is also known as the trachea. As it continues to flow down, it reaches the bronchial tubes, which branch out into both the left and the right lung, located on either side of the breastbone in the chest cavity. These lungs, between the two of them, are divided into 5 main sections or lobes. The lungs are also responsible for removing carbon dioxide from the blood and adding the oxygen to it. So if we were to direct our attention over here to the right-hand side, you can kind of see here that this is the bloodstream, and this is a structure called the alveoli. So we made it here into the lungs with the bronchial tubes. The very next structure and the part that's crucial in the gas exchange process is right here. This lovely picture that kind of looks like a bunch of grapes all clumped together. These are what we call the alveoli. There are thousands of these actually in the lungs, and they are basically tiny air sacs. Each one of these sacs is covered with a bunch of blood vessels that connect a system of veins and arteries that move blood throughout the body. When you take in a breath, the pulmonary, or the lung, that's what pulmonary means, artery branches out and brings blood containing lots of carbon dioxide and no oxygen into these vessels. Carbon dioxide leaves the blood and then enters the air, while the oxygen leaves the air and then enters the blood. Without these tiny little air sacs called the alveoli, we would not be able to survive because we would not be able to make this crucial exchange of carbon dioxide and oxygen. So a reminder here, a nice little breakdown. This would hypothetically be our bloodstream, which would be supplied by these little blood vessels that kind of envelope the outer sides of the alveoli. And the part that actually encompasses the alveoli is where that gas exchange happens. So again, the carbon dioxide starts in the bloodstream after being collected from the rest of the body. It then filters into the alveoli, which then is exhaled because it's a waste product. And then when you inhale, you bring oxygen in, it goes to the alveoli, where then it's transferred to the bloodstream and then pumped to the heart, so then it can be pumped to the rest of the body and then oxygenate your cells. And this is a repetitive cycle. So the big reason that I really wanted to review the major structures of the respiratory system with you is because I wanted to refresh your memory about the alveoli, and really they are going to be the main structures of the respiratory system that we're going to be focusing on as we talk about pulmonary surfactant. And one big thing, or one big force that we really need to take into account, is surface tension. Since the alveoli have their walls lined with a thin layer of water, this creates that force of surface tension. Surface tension actually tends to collapse these pulmonary alveoli, and as you can imagine, this can be a very big problem. If they aren't able to reinflate, then you won't be able to breathe in. Luckily, alveolar cells have found a way to counteract surface tension by producing a surfactant, which is a phospholipid protein that reduces the surface tension, keeping the alveoli open so that we can breathe properly. That being said, if we were to take a big, deep breath in, we can start to dive into the physics of surface tension a little bit. Don't worry, we won't be here too long. I just want you to have a very good understanding of why surface tension is a big deal in this process. So if we look at the middle of the screen, we can actually see water molecules, known as H2O, that stay very close together because of hydrogen bonds that form between the negatively charged oxygen ion of one molecule and the positively charged hydrogen ion of another molecule. You can also see this relationship in either corner in the upper hand side of this slide. Within the bulk water, which is back to the bottom middle of your screen, the molecules are equally pulled in every direction by neighboring molecules, so the resulting net force is zero. However, when you add air into the mix, the whole system becomes very unbalanced because at the water-air interface where they meet, water molecules are not surrounded by other water molecules. This creates too many cohesive forces between water molecules at the surface that pull the water molecules at the surface closer together, making the surface of the water shrink to the minimum surface area possible. So basically it's condensing in because these bonds are so tight. Now, the interior of the alveoli are also spherical, as we saw in the pictures and you can see kind of throughout this slide. So the net force of surface tension is directed to the center of the alveoli, which tends to collapse the alveolar walls towards the center. The magnitude of this force is actually predicted by the Laplace law. You'll see that, let me grab my laser pointer. You'll see that right here. Like I said, we're not going to spend too much time on it, but I really want you to kind of understand why this is a big deal. The Laplace law states that pressure collapsing the alveoli is directly proportional to the surface tension being generated by the molecules of fluid lining the alveoli and is inversely proportional to the radius of the alveoli. So basically, in a nutshell, the smaller the alveoli, the larger the collapsing pressure. Now, when we breathe out, as you can see down here, ta-da, the alveolar size decreases because the air that was originally inflating it has now been expelled from the lungs. This means that the alveolar radius reduces, which increases the collapsing pressure according to our Laplace law. It also means that for inspiration to occur, which is literally just trying to reinflate the alveoli, it will take too much force to overcome this collapsing pressure. It's just too much to go against, meaning that the entire inspiration process will be quite difficult. When it's difficult to reinflate these individual alveoli, it results in breathing problems because the alveoli cannot stretch back out to its full capacity. Luckily, this doesn't happen in normal individuals because human alveoli synthesize this lipoprotein called pulmonary surfactant. So what really is pulmonary surfactant? Surfactant is what lines the alveolar walls over the water film, which reduces the surface tension and counters the collapsing pressure that we talked about in the previous slide. Now, if we were to take a closer look at the alveoli, we can see that there's two main cells that make up its surface, type 1 pneumocytes and type 2 pneumocytes. Pneumocytes just mean lung cells because pneumo just means relating to the lungs, cyte just means cell. So put it together, pneumocytes, lung cells. And as we see here, they both make up the surface of the alveoli. And if we were to focus on type 1 pneumocytes first, we can see that they're the flat squamous epithelial type cells. These cells are very thin and widespread, making up about 97% of the whole alveolar surface, which helps ensure an efficient diffusion needed in the gas exchange process between the alveoli and the blood in the surrounding capillary network. Now, next to type 1 pneumocytes, we can see type 2 pneumocytes. These are more cuboidal shaped cells. Cuboidal just means that they're more cube shaped. You can hear that in the name. These type of cells actually contain something called lamellar bodies. These organelles are what secrete the actual pulmonary surfactant. In humans, lamellar bodies start producing surfactant at about 24 to 28 weeks in pregnancy. So as you're a baby in the womb, between week 24 and 28, that's where the initial beginning stages of pulmonary surfactant is being produced. Then at around week 35 of pregnancy, the alveoli have enough surfactant to keep them from collapsing. Now, let's have a look at the pulmonary surfactant itself. If you direct your attention over here. Ta-da! Pulmonary surfactant is actually a phospholipid protein. So direct your attention here. It's actually a phospholipid protein complex consisting mainly of a compound called DPPC. And I'm not even going to try to enunciate that. That'll be your challenge for after this presentation. Go ahead and look that up and then try to enunciate that. It is very long. So if we were to look at this DPPC, we can also see that it is accompanied by some surfactant proteins, which are marked A and D. But mainly what we're going to be focusing on is the DDP or DPPC, because that is what gives the main role of the surfactant that we're studying. So if we see here, this is what we call an amphipathetic compound. That just means it's a structure that consists of a phosphophilic or hydrophilic phosphate and an amine group attached to two hydrophobic 16 carbon saturated chains. So if we look here, this is our phosphate group. Hydrophilic just means water loving. And then we have our hydrophobic tails, which means that they do not like water. So they try to stay away from water as much as possible. The hydrophilic end then obviously adheres to the surface of the water film that lines the alveolar wall, while the hydrophobic tail floats in the air towards the center of the alveoli. So if we see here, the orange is that actual pulmonary surfactant. And here's a zoomed in version of our little DPPC complexes. You can see that the heads of these complexes are against that blue water line, while the tails are pointing inward. These hydrophobic tails here, as you can see, have very little intermolecular forces, so they don't really attach to one another. This considerably reduces the alveolar surface tension all over the surface of the alveoli. The protein component of the surfactant plays an immune role, such as helping with something called opsonization, which is basically where they just tag bacteria that invades the alveoli and has macrophages target them and destroy them. This also lowers the surface tension and brings in many benefits to it. It improves the compliance because it makes it easy for the alveoli to expand during inflation. During inspiration, the alveoli inflate and become larger in size. At this time, the surfactant becomes more scattered over the surface of the water film, making its amount relatively decreased as it becomes less dense on the water surface, which then reduces its action. This fairly increases surface tension, which slows down the rate of alveolar expansion, increasing the tendency to recoil instead. On the other hand, when alveoli deflate during expiration, meaning that the alveoli are decreasing in size, the surfactant starts to be clustered together, making it more dense over that water film, thus increasing its ability to counteract the surface tension. Phew, that was a lot of information on the last slide. But the biggest takeaway is that pulmonary surfactant actually counteracts the surface tension that is applied on the alveoli. So the more surface tension there are, the harder it is for the alveoli to reinflate when we inhale. And with the presence of pulmonary surfactant, it disperses that surface tension, decreasing the pressure put on the alveoli, making them more elastic and compliant. And that actually, in turn, just makes it easier for the alveoli to reinflate during breath in or inspiration. So let's compare what it would be like with the presence of pulmonary surfactant versus without the presence of pulmonary surfactant. So if we look on the left-hand side, the pink, you can see that we have, let me get my laser pointer, we'll call this alveoli 1 and alveoli 2, and then same over here. But right now we're going to focus on the surfactant incorporated. So in this particular comparison, number 1 has less surface tension, meaning that it has more surfactant per area. Both 1 and 2 have equal pressures due to the surfactant, and 1 will inflate faster than 2, even though, or at least until they're equal in size. If we come over here to the alveoli without the presence of pulmonary surfactant, we find a very different situation. Alveoli number 1 and 2 have the equal amount of surface tension. And number 1 over here has a higher pressure that it's fighting against due to its smaller size, as we kind of talked about with our Laplace law. The smaller the radius, the more pressure it has put upon it. Number 1 here also is more likely to collapse, meaning that it will be harder for the person to take air into the alveoli because it just can't expand to that full capacity. This situation here is what it's a big part of people who have major breathing problems due to pulmonary surfactant. Typically speaking, it's because they have a deficiency in pulmonary surfactant is where we see the major issues. Now that we've gone over how pulmonary surfactants are important to the breathing process, I think it would be good to discuss a little bit of the associated disorders and diseases that could take place in case a deficiency or abnormality takes place in the pulmonary surfactant production. And you can kind of see on the left-hand side of the screen, there is a list of disorders and diseases that can affect either adults, children, or both. One in particular that I want to touch on is the neonatal respiratory distress syndrome. For those of you who are curious, the acronym for that would be NRDS. Basically what happens is when a baby is born extremely early, also known as prematurely, they usually don't have enough pulmonary surfactant produced to keep their alveoli from collapsing during the inhale or inspiration portion of the breathing process. These newborns typically present difficulty breathing, which can be seen by either chest wall recessions or fast breathing rates, usually around 60 breaths per minute. This happens because breathing in takes too much effort to inflate the surfactant depleted alveoli because they are less elastic, or more technically speaking, less compliant. After difficulty in breathing, other signs could include poor tissue oxygenation. Basically, there's not enough oxygen being transferred to the bloodstream and then circulated to the cells of the body. This is what's known as hypoxia. Babies can also develop a fast heart rate due to this, and something called cyanosis, which is a blue discoloration of the skin. This disorder can even be deadly. Caring for such newborns involves treatments like ventilation support to help supplement an excess of oxygen, and delivering exogenous or artificial surfactant, which is mainly derived from the lungs of other animals. Now, before I conclude this presentation, I think it would be a good idea to kind of touch on a few current treatments and research projects that are going on regarding pulmonary surfactants. One company in particular I'd like to touch on would be Therapeutics. It's a Purdue University related startup that's attempting to create a treatment for the life-threatening lung condition known as acute respiratory distress syndrome, also known as ARDS. About 220,000 people in the United States alone are diagnosed with ARDS, and this disease can lead to organ failure and other health problems. And unfortunately, in this day and age, about 40% of people diagnosed with this condition often die. So Therapeutics is attempting to make a treatment using a nanoparticle that will mimic the behavior of natural surfactants in the lung. Basically, it will assist the body in the inflation and oxygenation process until the body can produce its own natural pulmonary surfactant to take its place. However, nanoparticles aren't the only method of treatment that's been attempted for replacing pulmonary surfactants. Actually, a journal article that was written in 1998 by John Gorek, I hope I'm saying that right. His journal article is called Pulmonary Surfactant Functions and Molecular Composition. In this article, he talks about how they've extracted pulmonary surfactants and also injected pulmonary surfactants into and out of animals, particularly rabbits and preterm lambs. And they've been having great success. Back in 1998 is when the article was actually written. And by that point, there had already been about 40 years of research. So now add that into 2021, and there's a lot more options. But in his article, he talks about how successful the animal trials have been. And I highly recommend this journal article for after you finish this presentation. There's a lot of good facts, a lot of more description in what we've discussed here. This is more of an abbreviated discussion on pulmonary surfactants, and the journal article that I'm suggesting goes way more in depth for those of you who are curious. These are some of the journal articles and resources and references that I used in making this presentation. Quick note for those of you who are curious, the article that I was suggesting would be this one right here. This is the one that was made in 1998, and it talks about the surfactant animal trials, or also the artificial surfactants. And for those of you who are curious to read about the nanoparticle approach, this would be the resource to look up. Here is the second page of references that I use. I just talked a little bit more in depth than I typically do in these type of presentations, so I wanted to make sure that I gave credit where credit is due. And there you have it. Thank you guys so much for watching my presentation. I hope, again, that you found it informative and that you learned a few things. If you have any questions or inquiries or something that you just wanted to ask, put it in the comments below and I will try to get back to you. Again, thank you, and I hope you all have a beautiful day.